Hi, I'm Owen White. I'm a neurologist at Royal Melbourne Hospital, or more specifically a neuro-ophthalmologist. I work with patients who have visual problems and also eye movement problems, and that's my particular research interest. I work very much in conjunction with the multiple sclerosis unit here at the hospital, as many of those patients have visual problems and eye movement problems, either causing double vision or jumping of their eyes. The reality is that many patients with many diseases are affected by this, and as such I've developed a research interest in that area and have a neuro-ophthalmology or eye movement laboratory working as part of the Melbourne Brain Centre, which is part of Melbourne University. Our patients are basically derived largely from the multiple sclerosis unit, and we've been doing this work for almost 20 years, although it's taken off particularly in the last year or so with the joining of people from Monash University to a lesser extent, but people working mainly here, particularly Lynette Mellist, who works in the laboratory, and Joanne Fielding, who has become a valuable research collaborator. The problems that we confront are the fact that the neurological examination really hasn't changed since the late 1800s. Many people have the impression that the brain is only using 10% of its capacity. The reality is that we actually only measure about 10% of the capacity of the brain with a conventional clinical examination. By comparison, imaging, MRI and other forms of electrophysiological testing have improved quite dramatically and give us a much better picture of the structure of the brain. My question was, how can we possibly examine brain function any better? Everybody thinks of the brain as having centres. There are centres for moving the arms, centres for moving the leg, centres for vision, centres for speech. The brain, in fact, works on a much more complex basis. Its main role is cognitive function. That means that we get information in, what's so-called afferent information, and we process that information to produce some sort of output. In many cases this may be speech, in some cases it may be movement, in some cases it may be preventing ourselves from falling. One of the first systems to develop in the body is the oculomotor system, and this is dependent on visual information coming in and ultimately either stabilising the eyes or moving them from one point to another, and all sorts of decisions need to be made in processing that information. Our laboratory looks at how we process that information. We look at what is essentially a network of different functions, all of which interdigitate, much like the London Underground works. If you think of that map of the London Underground you always see, you've got different lines, the Northern Line, the Central Line, the Metropolitan Line, all of which go from one place to another place, but all of which mix and match with other lines. If information slows along one of those lines, you may either get there late, you might get to the wrong place, or you might not get there at all. And what we do is measure eye movements as the output of defined visual information. We've particularly had some success at looking at multiple sclerosis, which is in many ways a shotgun disease. The unit here at Royal Melbourne has a very large research component and have been very supportive of all of the activities in our laboratory. What we have been able to show is that by looking at cognitive function in eye movements, we can actually detect abnormality in function when almost no other clinical evidence is available, and the patients may only have had one episode of neurological dysfunction. We've also been able to demonstrate that over time there is an accumulation of deficit that we can measure in the oculomotor system, and we're in the process of trying to define whether or not this will let us measure people's responses to medication, whether it will let us measure their progression, and basically use this in a clinical sense. Currently, we have several projects. We're trying to validate whether or not we can show progression in patients and whether or not we can show the effects of therapy, both therapies that have been used for some time and also therapies that are being developed. We're trying to match this against MRI findings. We also have some studies looking at the effect of fampridine and some of the newer disease-modifying therapies. For the moment, however, we're continuing to gain as much information as we can about the function of the brain overall. This is valid certainly for multiple sclerosis, currently the main focus of our research attempts, but also valid in other neurological diseases such as stroke and degenerative diseases such as Parkinson's disease and Huntington's disease.